Bom, gente, boa tarde. A gente vai começar agora a sessão de vacinas.
Good afternoon. We're going to start now the session on vaccines, which is going to be moderated by me and by me.
Boa tarde a todos. Primeiramente, gostaria de agradecer o convite do Daniel e da Carol.
First of all, good afternoon, everyone. I would like to first thank the invitation by Carol and Daniel to moderate this session. We're going to start with Professor Silvia Boscardin. It is a great pleasure to introduce her. She is an associate professor and professor leader of the University of São Paulo, the Department of Immunology of the University of São Paulo, and she went also to Unifesp, where she had her master's and doctorate in immunology. She has a postdoc of the Laboratory of Molecular Immunology at the Rockefeller University in New York, and her research integrates immunology, parasitology, and development of vaccines, focusing on the antigens for dendritic cells and the development of monoclonal antibodies against parasite diseases like SARS-CoV-2, malaria, and Zika. So I invite Professor Silvia for her lecture on the generation of human monoclonal antibodies against Plasmodium vivax.
Hello? Okay, good. So, good afternoon, everyone. Rodolfo, thank you so much for the kind introduction. I want to start, obviously, by thanking Carol and Danielle for the invitation. It's really, really a pleasure and an honor for me to be here. This is my first time presenting in a national malaria meeting, so it's very significant for me. So as Rodolfo mentioned, I'm going to talk to you a little bit about a project that we have. It's still ongoing, and we have been working for the last few years on the generation of human monoclonal antibodies against the infectious forms of Plasmodium vivax. As I am in a table that we will discuss vaccines especially, right, I decided to tell you that the development of monoclonal antibodies against infectious diseases can actually help the design of more effective vaccines, because I won't talk about vaccines at all, okay? And why they do that? It's because by using monoclonal antibodies, you can identify protective targets. For example, you can identify the epitopes. You can, for example, accelerate responses during the pandemics, because monoclonal antibodies are normally a little bit easier and faster to produce than vaccines. So, for example, you can start using them in the context of a pandemic in the beginning, right, to treat people, for example, that are highly susceptible and that need treatment very quickly, or that can evolve to more severe disease. They are very useful for the identification of variants and for the identification of evasion of the immune system. And that what happened, for example, for the COVID-19 pandemics, when the antibody that used to neutralize stopped neutralizing, scientists figured out that probably the virus was already mutating. And also, they can be very helpful for immunogen design. So, for example, if you know your target, and if you know monoclonal antibodies that actually block that target and block the infection, for example, you can design antigens that can actually serve as vaccines later on. Okay, so I hope that I convinced you that monoclonal antibodies are good for vaccine development. Okay, so basically the main goal of this talk is going to, I'm going to tell you obviously about the development and characterization of human monoclonal antibodies against the Plasmodium vivax circumsporozoite protein. So as all of you know, circumsporozoite protein is the protein that is the major protein on the surface of the sporozoites that are the infective forms of Plasmodium. So to do that, we basically go to, so we have sort of a workflow that I'm going to go very quickly with you, just to let you know how we do it. So from the beginning until the actual obtention of the monoclonal antibodies. So basically, we have first to select the donors, right? So for this particular project, we selected people that had already infection with malaria, that were already infected with Plasmodium vivax malaria. Next, we get the PBMC, so the peripheral blood mononuclear cells of these people. And then we enrich these PBMCs for B cells, okay? And then to stain these B cells, what we do is that we use, for example, a protein, or the pathogen, the whole pathogen if you want, but a protein as a bait. Okay, as you know, every B cell that responds against an antigen has a B cell receptor that can recognize that antigen. So if you get a protein and you stain this protein with a fluorophore, and then you dump it on the top of the B cells, some of them will react with this protein and will be stained. And then we can sort them using flow cytometry, using a cell sorter, and then we sort each one of the B cells that interest us, or that are stained by the protein, into 96-well plates. And then what we do next is basically we get the RNA of these B cells, make a cDNA, and then we amplify by PCR the variable regions of the heavy chain and of the two types of kappa chains that are expressed in mammals and in humans, the light chain expressed in mammals and in humans that are kappa and lambda. Okay, so we amplify this, and every time we get a match in a way that we get an amplification of the heavy chain, and then an amplification of either a kappa chain or a lambda chain, we have a match, and we send these sequences for sequencing. And then we discover what is the V genes, the variable genes that are part of this B cell receptor. Okay, and once we figure out, like, we know that they can be produced. So what we do is that we clone the variable region of the heavy chain into a plasmid that encodes the constant region of an IgG1. Okay. And we do the same thing for the variable regions of the light chain. So we clone the variable region of the kappa chain together with the constant region of a kappa chain into a plasmid, and the variable region of the light chain into a constant region of a gamma chain, a lambda chain. Okay, and then we use this pair of plasmids to basically produce transiently transfect HEC293 cells, produce the antibodies. Then we fish these antibodies of the supernatant of the cells, and then we work on the characterization of these antibodies in vitro or in vivo using mouse models. Okay, so here we go. So for this particular project, so we had to get a bait, right? So the bait was basically the chimeric CS protein that was already introduced in the first conference by Dr. Irene Soares. And it's the one that is going to be used for the Plasmodium vivax vaccine that I think Dr. Ricardo Gazinelli will tell you a little bit about in the last talk, right? So, but just to introduce you very quickly, because this is going to be important information for what I'm going to say later. So this chimeric protein, it's a CSP that has repeats, the central repeat from the three allelic forms of Plasmodium vivax. So it has repeats from VK210, VK247, and P-Vivax-like, okay, and Vivax-like. And then we have in the N-terminal, a small portion at the N-terminal that it's highly conserved, and the whole C-terminal. So, and then we have, so once we got this, we decided that we use this chimeric protein as the bait. Mariangela came into play. So Mariangela is a very talented postdoctoral fellow in my lab, and Mariangela decided to take up the challenge and try to produce these antibodies, right? So then what she did, she got basically the sequence of this protein, codon optimized it to expression in XP293 T cells, and then she produced this protein fused with an AV tag that would allow us to biotinylate the protein and then come with a streptavidin stained with a fluorophore, and then basically stain the B cells. So this is just to show you that the protein was produced successfully, and then we had to go to the second part of the job, that was to basically select individuals that had previously malaria. And for this, we used a cohort of 32 individuals infected by Plasmodium vivax from the city of Mancio Lima in Acre. And this is a collaboration of my colleague, Dr. Marcelo Urbano Ferreira, at the Department of Parasitology. So Marcelo and I co-oriented a student that used basically this cohort, and we had samples from PBMCs from these people. Okay, so Marcelo was essential in this part of the project. So what Mariangela did, she basically got the plasma of these patients and ran an ELISA, a simple ELISA. So these are the titration curves of the ELISA, and here's the area under the curve. And she figured that out of these 32 individuals, only 6 individuals had let's say, antibody titers good enough for us to try to see if we could fish out the B cells. So we basically selected 7 donors, 25, 29, 34, 36, 57, and 60. 6 donors, sorry. Okay, so then Mariangela went to the Rockefeller University to work at the laboratory of Dr. Michel Nussensweig for a year. One thing that I forgot to say is that Mariangela has a poster in which she's showing this result, so she probably knows more than I do. So if you have any doubts, please go to her poster today. So she went to spend a year in Michel's lab, and she sorted these cells that are specific for CSP for all these 6 patients. So this is basically the gating strategy for you to see. So we normally remove other cells, we do CD20 to get the B cells, and then we do a double staining with CSP in 2 different colors just to improve the specificity. Good, thanks. Okay. So then Mariangela sorted all these cells, and then in the end, she ended up with 80 paired sequences, which I mean when I say 80 paired sequences, like heavy chain and light chains. Okay, and so those are the sequences per individual that she's plotting in these pie charts here. So we got, for example, for individual 25, 12 sequences, 29, 14 sequences, and so on. And then in the colored slices, we can see she's representing sequences that are clonally related, that probably came from the same B cell. Okay, so it's life, right? We are very variable. So basically we have individuals in which we could not get really good matches, but had only 3 and they were all singles. And we had individuals in which 71% of the B cells that we got specific for CSP were clonally related. Okay, so then she basically did an analysis of the variable regions. So the variable regions for the heavy chain, the most common was the V333, for the kappa chain, V228, for the light chain, V310, and those are actually quite common in Plasmodium infected individuals. Okay, when she went to analyze, for example, the number of mutations in these variable regions, because these variable regions are called variable because they vary a lot, so they have lots of mutations, right? So she could see that in average it was like around 15 mutations in the heavy chain, a little bit lower for the kappa and light, but none of these results were actually very different from what is seen in Plasmodium infected individuals, Plasmodium falciparum infected individuals, and in other infections. Okay, so this was okay. So then Mariangela picked 39 antibodies to produce. Okay, so she randomly picked 39 antibodies, 1 representative of each one that was clonally related, and all the single ones. And she produced these 39 antibodies. And then she did an ELISA titration of each one of these antibodies against the chimeric protein that Irene had given us, right? So then here you can see, this is the area under the curve, the same result as here, but here you can see as bars, right? And you can see that like some antibodies really bound well to this protein and some of them did not, right? But then we wanted to see, as we generated them against a chimeric, let's say, protein, we wanted to see if we could sort of discover which antibody was directed to which allelic form, right? So to do that, what we did is that we came back to Irene again, right? And then she gave us some constructs that she had previously produced in the past that were like flagellin, linked to either the variable regions of VK 210, 247, and Vivax-like. So she generated all these 3 different proteins that only have the repeats of VK10, VK247, and Vivax-like. And then Mariangela basically did I analyze, right? These are the curves, and in here I plot the area under the curve. So to our surprise, we got, I think it's 11 11 antibodies that would exclusively bind to the variant VK210, 2 antibodies that exclusively bound, not that well, but they bound to the variant PV-like, and no antibody that bound exclusively to the VK247. But it bound, for example, this one bound to 247 and a little bit to PV-like, and this one bound to all the 3 allelic forms. Okay? So then we had to really see if this antibody would do something, right, to the parasites. And then come into the picture Rogério Amino from the Pasteur Institute and Maísa Araújo from Fiocruz Rondônia, because Rogério has this huge expertise in sporozoite biology and antibodies against, and the description of antibodies that are cytolytic to these sporozoites. And Maísa is the girl that has, obviously, the parasite. So Maísa is the one that can produce P-Vivax sporozoites for us to really test, right? And Délio, thank you so much for really being into place, because I know that you helped a lot too. Okay, so basically what we did here is, the first thing that they did was, so Mariangela in Kenya, that was at that point an undergraduate in my lab and now is a PhD student. They went to Porto Velho, and Rogério went too, and then they decided to produce the antibodies and then test the binding first, and then the capacity, the cytolytic capacities of these antibodies to either kill the parasites or induce the circumsporozoite precipitation reaction. And the way Rogério does this is by flow cytometry, so they incubate the sporozoites that Maísa gets for us with the parasite, with the parasite with the antibody, and then we come 37, 45 minutes, and then an anti-IgG FITC, and to check for killing of the parasite, we add also 7-AAD, which is a DNA marker, right? It marks dying cells. So, and then we analyze this by flow cytometry. And what they could show is that, so this is the antibody, each antibody, and this is the percent of sporozoite-specific green, percent of binding of the antibody to the sporozoites. So we could see that, so here are the antibodies that do not bind, and there are some of them that really bind well, and some of them that don't bind that well, but are above the, let's say, the negative control, which is here. If we look at the median fluorescence intensity, we can see that the ones that are in dark blue here, they actually vary the intensity, so they're binding, but some of them are not binding super well, but they're there. So we got 16 that bound and 7 with a higher MFI. Okay, so then what they did is that they did an immunofluorescence and indirect immunofluorescence to really see if these antibodies would bind to the sporozoites live, right? So you could see here that they do bind to the sporozoites in different, let's say, patterns. We have still to repeat these experiments to see if those are really patterns that are reproducible or it was just a problem with the assay. And we found a strong correlation of these antibodies that bound to the P-Vivax sporozoites to the binding of the VK210 variant. So basically, probably the patient was infected with this allelic form, and our antibodies really bind to that. Okay, so the next, what they did was to really check if this antibody could bind to sporozoites and trigger the circumsporozoite precipitation reaction. And actually, our favorite antibodies, they could do it, but then something different or interesting happened. Some of the antibodies that wouldn't really bind that well also could induce the CSP precipitation reaction. So they're binding somehow, but not probably with high affinity. And this is just a correlation of the antibodies that really bind to sporozoites and also are able to do the CSP reaction, to trigger CSP reaction. So these are the good ones, and these ones are those ones that do not really bind that well, but actually trigger CSP reaction. And to finish, I still have 1 minute. So, and finally, what they did was basically to check killing. So if these antibodies would be cytolytic. And the antibodies that are in dark green, the ones that bound, it was interesting because some of them really didn't, like they bound with, like the percent of fluorescence was small, but they couldn't really kill more sporozoites than some of them that had a high percentage of fluorescence that would really bind better to the sporozoites. And obviously the ones that didn't bind could also kill. So this is interesting. We're going to have to really pay attention to this phenomenon to see if it's some kind of problem with the assay or if it's true, okay? So, and finally, Rogério, we shipped these antibodies to Paris, and then Rogério, with the help of his student Fiona Davis, what he did is that he tried to use these antibodies in the chimeric sporozoites that are P. Bergiae that express the VK210 from Plasmodium vivax. And this for us is really important because we want to go in vivo to see if we can block infection. And to our good surprise, all our antibodies that were able to kill Plasmodium vivax sporozoites were also able to kill the chimeric parasite Plasmodium bergiae, Plasmodium vivax sporozoites. Okay, so the take-home message is that we could generate 39 monoclonal antibodies against Plasmodium vivax CSP. 11 monoclonal antibodies were specific to repeats from VK210 variant, and 3 to the P. vivax-like variant, while none showed exclusive specificity for VK247. 12 mAbs bound to the sporozoites of P. vivax, including inducing CSP reaction and mediating parasite killing. There was a correlation between sporozoites recognition by mAbs and induction of CSP reaction, as well as parasite killing. And this phenomena were similarly observed using both P. vivax sporozoites and the chimeric P. bergiae P. vivax CSP sporozoites. And the next steps, we want to perform in vivo protection experiments using PBPV sporozoites. We need really to map more precisely the binding of these monoclonal antibodies, and we want to try to improve monoclonal antibody affinity by site-directed mutagenesis on hotspots to see if we can get better antibodies. So last but not least, I wanted to thank again Mariângela Silva, Kenia Abreu, Marcelo Urbano Ferreira, Natália Soares Ferreira, Márcio, and all the students that are now in my lab, Michel and his team at the Rockefeller, Irene and Tatiane Rodrigues de Oliveira at the Faculty of Pharmaceutical Sciences, Rogério Amino and Fiona Davis at the Instituto Pasteur, and Maísa da Silva Araújo and Najara Akira Costa dos Santos at the Fiocruz Rondônia, and for this particular project, primarily FAPESP, CNPq, and the University of São Paulo. Thank you so much. Time is over.
I would like to invite Dr. Patrick Duffy to join us. Dr. Patrick is a physician and leading researcher at the National Institute of Allergy and Infectious Disease, part of NIH. He actually served as chief of the Laboratory of Malaria and Immunology, and also chief of the Vaccine Development Unit, and his work focused on innovative malaria vaccines, strategies, preclinical evaluation of candidates, clinical trials, and development of immunological, molecular, and entomological assays. The title of his presentation today is Vaccine for P-Vivax Discovery and Testing. Please welcome.
Thank you. Thank you, Daniel and Carol. I'm not sure where you're at. Oh, there you are. Hi, Carol. Amazing venue, amazing meeting. It's really been awesome. You guys did a great job. This is a story about Vivax, but I'm going to start with falciparum because it gives the context and sort of the rationale. And I advance here. So this is a simplified life cycle so that people are all on the same page about what vaccines can do. And our flagship program is to make a vaccine that kills parasites inside the mosquito. And that's useful for malaria control and elimination. We do work on anti-infection vaccines. It's not our flagship program, and we're delighted to partner with other groups that are making pre-parasitic vaccines or anti-infection vaccines to combine with our transmission blocking vaccine, which is what we really enjoy doing. But we really enjoy working on whole sporozoites also, another form of anti-infection vaccine. You learn a lot with those. But the idea here is to combine a transmission blocking vaccine, one bottleneck in parasite development, combine it with an anti-infection vaccine, the other bottleneck, and by doing that, interrupt malaria transmission at one of those critical points. We also have programs, particularly on placenta malaria vaccines, very feasible, should work, hasn't worked yet, but we know we're going to get there, and we're following the field in severe malaria and trying to learn more about that. So I mentioned TBV, transmission blocking vaccine, is it's our flagship program. When I got to NIH to lead the program in 2009, this was the program we prioritized, partly because nobody else really was investing in the clinical development. And we really needed a clinical development path, and I feel like that's what we've contributed to the field. So this is a membrane feeding assay. This is one way that we can measure the serum activity that we generate with our vaccines. We add sera to parasites in that feeding device, and that way, if we compare sera before vaccination to sera after, we'll see if we're reducing parasites that transmit to mosquitoes. And that can use either cultured parasites, and that's a standard membrane feeding assay. We do that at NIH, or we can use parasites directly from infected people, and I'll get back to that in this moment. A week later, you dissect the mosquitoes, and those pink orbs are the oocysts, and that's a way to quantify the level of infection. Actually, quantifying the level of infection is a more reliable endpoint to compare assays than how much you reduce the proportion of infected mosquitoes. Then this is what you can do in the field. It's a very old assay. It's a xenodiagnostic to tell you whether there's transmissible parasites being carried by somebody. It's, we call it a direct skin feeding assay. And what you can see is that there are 2 cups here on this study participant's arms. Each cup has 30 mosquitoes in it, and the mosquitoes will feed for 15 minutes on their skin. And our standard practice generally is every 2 weeks. They come in, we don't check a blood smear. They're infected so frequently in the communities where we work, and we see whether anybody transmits. Again, a week later, we dissect to have the endpoint, and that way we can compare vaccinated people to comparator groups. And even though this has been around a long time, what's different in Mali, our partners, is we worked with them and really scaled this up to a level where you could get enough endpoints to do efficacy trials. So for example, Mali can do the direct skin feeding with 60 mosquitoes on 400 people each week, which requires 24,000 female mosquitoes. And then next week, they get dissected while another 24,000 female mosquitoes are feeding on people in the field. So it's quite an operation, and they're really remarkable teams there in Mali. And we just published our experience over 15 years together with our colleagues in Mali, and that included over 38,000 of these assays. Very safe, very well tolerated. We use mosquitoes that are common in those areas, and there's evidence that when you're frequently exposed to the same mosquito, you develop tolerance to the effects of the mosquito bite. So our lead candidate is PFS230D1. PFS230 is a large, complex, cysteine-rich protein on the surface of gametes, both male and female. So we're targeting the gametes with this candidate. We compared it to other candidates, both in our group and other groups. This is our lead candidate now. And to do those studies, we used the membrane feeding assay that I showed you earlier to measure the serum activity. Since then, since that's 2000, and we started that trial in 2015, I think, at the NIH Clinical Center. That was a study at the NIH Clinical Center. That's why we used that assay. Then we went to Mali. We tried different adjuvants, a hydrogel AS01. We're now working with Matrix M. And when we moved to Mali, then we moved over to this additional assay so that we could measure what we call efficacy. This is a surrogate endpoint of efficacy that you can use to predict what clinical benefit in the community will be. So one of the important trials that we did was using AS01 from GSK, and what I'm showing you here are the endpoint of infected mosquitoes. So what you're seeing in the left panel is the comparator group. What you're seeing in the right panel is the PFS230D1 group. And each column is a month, how many infected mosquitoes were produced by feeding 60 mosquitoes. This is 200 school children in each arm. So 200 school children in that left arm generated about 100 infected mosquitoes in that first month of the first season. And here's the second season of follow-up. And you can see this profound effect that the vaccine had on the field. So it really has great efficacy to reduce infection of mosquitoes, an 87% reduction in the proportion of infected mosquitoes. Now, the intensity of the infection, which is an important predictor of whether a mosquito will transmit parasites either to an animal or a person, that's indicated by the intensity of this red. And so we pretty much eliminated those heavily infected mosquitoes, which we think will have a separate effect from the effect of reducing infected mosquitoes completely. So where we're at now with this program is we are on a development path for licensure. We're combining our PFS230D1 vaccine with R21. R21 is one of the 2 licensed malaria vaccines that are being deployed in Africa. It's made by Serum Institute of India. And so we're partnering with them to develop. This is a partnership between ourselves. We have a network of sites across Africa headquartered in Bamako, Mali. And then the vaccine we developed, and I should mention, Adrian Hill at Jenner Institute is the one that developed the R21 vaccine and licensed it to Serum Institute of India. And why would you make a vaccine like this? Well, these are the mathematical modeling results that tell us what we should expect. This is Joe Challenger at Imperial College. That's who we work with, him and Tom Churcher. This is the cumulative incidence of clinical malaria in the Bamako region of Mali, where we've been doing all of our studies. And so this is, let me advance this. This models a primary series of PFS230D1 at zero time point, a booster dose at the beginning of the next year, no booster dose at the beginning of the 3rd year to see if any benefits remain there. So it's really a primary series with 1 annual boost, but you could continue to boost. And this uses the mathematical model at Imperial, and it assumes 80% coverage. The red line is just receiving standard of care, which is pretty much bed nets in the area. That's the cumulative incidence per 100,000 people. And so you're seeing that the average person has had 5 episodes over that period of time. If you add R21 alone, that's the purple line. If you add PFS230D1, it's this line. That's based on the phase 3 data from R21, and it's based on the phase 2 and adult phase 1 data that we gave to Joe. And if you combine them, what you can see is at the end of the second year, you've reduced the cumulative incidence of clinical malaria in those communities by about 80%. Malaria, as you can see from the numbers, is extremely intense. The average person has 5 clinical malaria cases and a lot more parasitemias in those areas. But the bottom line is P-Vivax VIMT is needed for control and elimination of Plasmodium vivax in areas that particularly want to eliminate. And so that's where the Vivax story begins. And once we had seen good results in animals with the PFS230, we committed ourselves to starting to develop a PVS230D1. But you'll note that started in 2015. And then we got resources so we could manufacture this in 2018 to 19. And then we had a delay because of COVID, and we couldn't do trials in the NIH Clinical Center because they didn't want people coming into the clinical center. And so we ultimately, and I'm showing the leaders in each of these areas from our group. So Joel Goldberg led this study, and we just did a dose escalation that was staggered for safety to know that the lower dose was going to be safe to move on to the next dose. And it was 5, 25, and 50 micrograms, but all of these were in 50 micrograms of the Matrix-M adjuvant, and these were the antibody titers that we've seen in this first in-human trial of PVS230D1. And what you can appreciate a little bit is that the low dose of antigen worked just as well as the other doses. That's good for us. It gives us some space to operate when we try to combine this with other partners. And I'll mention also that the titers that we got with PVS230D1 look pretty similar. It's a different antigen, but it looks pretty similar, so we feel pretty good about that. But then how do we measure the activity? We can culture P. falciparum in our lab and do the feeding assays, but we can't do that for Vivax. And so that led us on another path, which was a path to develop a source of P. vivax parasites that we could use for our studies. And so we started a partnership with James McCarthy, who's now down in Melbourne, but he's an amazing colleague, and he and I actually overlapped for training at NIH. James set up a P. vivax challenge model with parasites that an Australian traveler brought back from India. So this is an Indian isolate. And he's the one that established the P. vivax infection model. He also demonstrated that it was possible to transmit with these parasites with a limited number. And John Woodford is the amazingly talented young physician scientist who worked with James and did some studies with P. vivax and then spent 2 years with us to help us set up this model. And then our clinical leader of the first study, which was just to lay down cell banks, was Dave Cook. And you can see 2 participants that came into the NIH Clinical Center. We inoculated vials of the cell bank that James had given us. He gave us 5 vials. So they were very precious to us. Fortunately, the first 2 went to good use. The arrows show where we collected blood from these individuals. And we then got FDA approval for that, one of those cell banks, to now inoculate them into other people. And so FDA has approved this, and that allowed us to move forward then to doing studies with our own cell bank, inoculating that into people. And this is the parasitemia that we see. Day 0 is the day of inoculation. And you can see the steady increase in blue of the parasitemia. That's parasites per ml using a qPCR. And for the PVS25, that's just copies per microliter. We need to standardize that so we can quantify still. But you can see the basic pattern. And the dashed line is when we treat people. They usually feel pretty sick by the time we treat them. But we have to take them that far because when we do the feeding assays, they really uniformly transmit on day 11. That's a pretty standard thing that we've seen. This is, I think, data from the first 8 people. We've now done 14 people with this assay. The other thing that was a little bit unexpected based on some previous data, but clearly the direct skin feeding assay, the blue line, is more sensitive for detecting transmission than the membrane feeding assay. An important reason to use the DSF for your efficacy endpoints. So this platform, Controlled Human Malaria Infection and Transmission Platform, generates gametocytes that we can use for our studies. That was the primary initial impetus for this. But then it has a lot of other benefits. So, well, this is the first one that I talked about. These are the titers. This is the functional activity using this assay. There's something called transmission reducing activity on the left. That's the one I said is the reduction in parasite levels in the mosquitoes. And then the degree to which you reduce the proportion of infected mosquitoes is the transmission blocking activity. So this told us we were generating good and significant functional activity in the people that received the vaccine. And it was similar at the low, intermediate, and high doses. And we've been very blessed to have a partnership with Maisa and Delio, and Maisa has been doing parallel assays using field isolates, and she's completing the data set, but we know that she's completed the low dose, and it's a very similar kind of TRA, transmission reducing activity, that we got. So the DMFA at both sites showed that this vaccine induces serum functional activity. Now, there's a lot of other uses for us. So this took a long time for us to set this up. Probably took James and I about 10 years to get the resources and things to do it. But it was waiting forever, and then everything happened all at once. So first of all, we started concentrating the gameta sites to try to improve infection. And what you can see is that when you concentrate gameta sites, that's the squares versus the whole blood. Not surprisingly, you have more parasites in your sample, and you get more infected mosquitoes. And you also get more heavily infected mosquitoes. And that's useful. Those sporozoites are really useful to us, and we're in a program now to develop the cGMP procedures to use the sporozoites we're getting out of our volunteers and put them into Cineria's mosquitoes so that they can do cGMP manufacturing. And soon, hopefully, we'll have cryovialed PVSPZ that people around the world could use. You got to ask Cineria, though. To infect people. But there are people all around the world infecting with PFSPZ, so let's be optimistic about what's going to happen. So enriched gametocytes yield heavily infected mosquitoes, and these are now suitable for cGMP manufacture, and previously it was too low to consider that. We also have used the sporozoites to infect monkeys, and what I show you here are data from 5 Aedes nancymae and 3 Cimeri boliviensis. And this is to show that all of the animals became infected. This is early data, but just to show that all of them had a pre-acidic phase, and then from the time that we started doing the PCR, had detectable parasitemias. We do see a higher parasitemia in the Aedes than the Cimeri, you can see by these numbers. And so the sporozoites are healthy, and they uniformly infect. And now with the NHP model, there's other things that we can do. One of the things I mentioned is we do need a CSP partner for our PVivax transmission blocking vaccine. And so this is a way that we can test preclinically our different candidates. Again, if somebody else has a better CSP, we're delighted to work with them rather than going through all that work ourselves. We're interested in relapse. And this is the data I was trying to remember, but I finally got the data. I was trying to remember for George to talk to him about it. But what you can see here, 3 of the Cimeri that we infected, here are their parasitemias. It was a dose of 10, 100, 500. We followed the animals for a period of time. 2 of them lost their parasitemia. We splenectomized because we're interested in the spleens. We did not see that the parasitemia went up as we thought we would. We saw a few sporadic infections about 60 days after the original inoculation, and then we treated the animals with mefloquine. And then after treating, about 2 months later with mefloquine, we saw that all 3 animals had parasitemia again. It was very brief, but all of them had it around the same time. We're suspicious that those were relapses. We've subsequently treated these animals with artesunate. We've euthanized them, and we've collected the liver so we can look for parasites there. And then also we're interested in the spleen. We're doing human studies. We've submitted a protocol to do imaging. This is imaging that John Woodford did with James McCarthy back in Brisbane. And so we'd like to repeat those, but we have the monkeys as sort of our histopathological correlates that we can do for what we see that looks similar between monkeys and humans. And of course, we need to use this for pregnancy malaria. So we have a model of placental malaria that we developed. It's the first really plausible model of placental malaria in an animal. It uses Aedes nancymae. We pair them ourselves in our research facility. And then after pairing them, we wait till they become pregnant, and then we explore what we first did was P. falciparum, and P. falciparum in these monkeys looks just like what we see in pregnant women. P. falciparum mature forms accumulate in the placenta, where you'll see just scant ring stage in the peripheral blood. The binding in the placenta is always to chondroitin sulfate A, but in the peripheral blood, it could be CSA or it could be CD36. That's what we see in women also. And this shows antibodies, so they develop antibody titers. Those are the lines against FAR2-CSA domains, but also those columns show the functional activity. So we're able to induce this broadly neutralizing antibodies that protect women, we believe, over time. The interesting thing is you'll appreciate that the peak titers for FAR2-CSA early in the course of multiple infections with this particular monkey, they don't coincide with the activity. And then Justin Derechimo led a study with our group and our partners in Europe who had the first generation of FAR2-CSA vaccines to show that these, in the monkeys, just like in women that they tested, their first generation get only variant-specific and not broadly inhibitory activity. And finally, this is to bring all this together. Our next trial will actually re-enroll the people that were in our phase 1 study that we used the standard membrane feeding assay to give them a booster dose, and then we'll enroll a new group for a primary vaccine series. We'll use Matrix M, we'll induce protective immunity, and then we'll be able to do the direct skin feeding to show whether the vaccine has prevented them when we infect them. So we'll infect them here after the vaccination, and that'll allow us to see whether they're transmitting parasites to mosquitoes. And because it's such a reliable model, these are the power calculations. 10 people really gives us over 90% power to detect a vaccine efficacy of 60%, and I told you our falciparum is about 85 to 90%. And we have been having discussions with Maisa and Delio about how we could move forward with setting up this similar platform to assess this unusual kind of vaccine, and then to do a field trial together. And I should mention that both the R21 and the PFS230D1 for falciparum can interrupt transmission, so our assay, by feeding on people, will tell us the activity of each of those vaccines to reduce transmission in the community. So the platform supports numerous projects that I told you about. It generates gameta sites that we can use immediately for our studies. It's generating sporozoites that we can do monkey or human studies once we have the cGMP manufacture and FDA approval for the PVSPZ. And our monkey, in parallel with our humans, will give us a good opportunity to find correlates that we can then do more mechanistic studies in the monkeys. And a VIMT will be useful for P-Vivax elimination programs. And thanks for your attention.
Thank you for your wonderful presentation. Agora, gostaria de convidar a professora Luzia Carvalho, doutora Luzia Carvalho.
Now I would like to invite Dr. Luzia Carvalho.
Mas eu nem apresentei ela, vocês já estão batendo palma.
I didn't even introduce her and you're already clapping.
Luzia é pesquisadora titular da Fiocruz Minas.
Luzia is the full researcher of Fiocruz Minas. She was the Vice Director of Research and Innovation at Fiocruz Minas as well. She's a pharmacist. She had her master's and doctorate in Federal University of Minas Gerais. She also did a doctorate in New York, and now she coordinates the group of molecular biology and immunology of malaria. Her research focuses on the immune response and inflammation and also the host-parasite relationship. Thank you, Luzia. Thank you.
The organizers, special Carol and Daniel. This was lovely meeting, and they are so sweet. So thank you very much. And it's special because they bring a lot of students here. So this is very important in Brazil. Also, Josué helped to bring the students. So thank you very much also, Josué. I will talk about this. I don't know if I go play for the, Fabio told today the Vivax make the sick game. I think the point has a sick game, if you ask. But the guy from the desk told the computer has 3 problems: hardware, software, and peopleware. Perhaps we are in this peopleware. So let's see. Oops. Okay, this will be too much for me. Okay, I will talk here about this. Oops. Is a problem of the peopleware. So I will talk about this long-term follow-up study of the Plasmodium vivax Duffy binder protein. I think the DBP, the DBL domain, the region 2 of the DBP is the more persistent vaccine candidate. We're still not there because we have a hard time to make the vaccine, the people that has been trying to work with DBP. But this is very important for the parasite. I think because there is a lot of students here today, I do like, I think that I don't know if I go to this mouse. This is too slow for me. Okay, so DBP, why DBP is important? Because DBP, most of the strain of Plasmodium vivax depend of the interaction between the region 2 of the DBP and the DAC receptor, the Duffy receptor in the surface of the young reticulocytes to invade the erythrocytes. And this explains why let's see. No, it's there. No. Okay. This explains why in Africa we have most of we don't have low levels of Plasmodium vivax because most of the people are DAC negative, but still have a lot of gaps in this because more recently we discovered that DAC negative people can infect with DAC. I think that for the students it's very important that this was DAC receptor was discovered by Lou Miller about 50 years ago. And also you cannot say these people here are not infected, they don't use the DAC to infect to get infection of Plasmodium vivax because you know that in the precursor in bone marrow they can express DAC. So you still have a lot of gaps there. Okay, what we have learned in the last 20 years about the antibody response against DBP. The idea of a vaccine against DBP is to block this complex interaction between the DBP and the DAC receptor. So we learned something that's very important, that DBP antibody response induce these antibodies, these are able to block this interaction. And this is in different endemic sets, include high endemic sets like Papua New Guinea and also here in Brazil. Also, what's important that inhibitory antibodies against DBP can confer some degree of protection against Plasmodium vivax. This was demonstrated first in Papua New Guinea and also for Marcelo here in Brazil. And more recently, last year, Chitney Chetan, he was a former postdoc of Lou Miller in this controlled human malaria infection, demonstrated that DBP vaccination also confers some degree of protection. In the beginning, phase 1 was very, oops. Okay, but there was some reduction in Plasmodium vivax multiplication, and this was mediated by antibodies. But as Fabio told, vivax is not stupid, so make this sick game. And the bad news is that very low people develop this strain-transcended inhibitory antibody response, and also in high endemic areas or low endemic areas like here in Brazil. So the problem is because DBP is strain-specific, and I think that we make the more convinced evidence, I would say, that Plasmodium vivax DBP induces strain-specific immune response because we study naive people during an outbreak here outside of the Amazon. Was a fisherman that came here infected with a relapse of Plasmodium vivax. So we have these naive people a short outbreak was about 20 people. Everybody was treated, but we have the opportunity to check what's the outbreak, the variant of the outbreak, and was a single allele of DBP that was associated with this outbreak. So here, co-cells express the DBP from the outbreak and SAL1, express SAL1. And when we incubate with the sera from the outbreak, you can see that they only bind to the homologous system. They cannot bind here the heterologous. So just to illustrate, this is prime infected people, so they have low titer of inhibitory antibody response, but you can see that they bind to the homologous, not to the heterologous. So yes, the DBP is strain specific point. And why part of this is because Chetan still, sorry, I have to say, try to make a vaccine with a single allele. It's not possible, I believe. So why polymorphism of DBP explain part of this. This was made during the PhD of Thais. Now Thais is here, present in the young investigator, make very good collaborations. So the DBP is important for the parasite, but the parasite is not stupid. So this is a weakly immunogenic protein, induces strain specific. But why nobody give up? Because there is a good news, because there is very few people that develop this strain transcend immunity, that some people call elite responder or persistent responder. So in high endemic areas or low endemic areas, there is people with this transcend immune response. And this suggests that DBP where is this? That DBP, that DBP presents some epitopes immunogenic that's conserved that you can target for this broadly inhibitory antibody response. This was the idea of John Adams. The second, I think the other also former postdoc of Lou Miller. So Shitney Chetan, John Adams, make a great contribution in the field of DBP. So the idea of this DECNU family, a synthetic antigen for that was approved of concept, was the idea here was delete this the most polymorphic epitope of DBP, the DEC. So Francis was at that time a postdoc of John, and they showed that mice, when immunized with DECNU, these mice induce a broadly antibody response, but mice are not human. When you check this, if the people here in the Amazon that has been long-term exposed to Plasmodium vivax and has broad antibody response against DBP, they don't produce antibodies against DECNU2. So came the second generation of this, that was DECNU2, that was much more mutation. Camila was a former postdoc in my lab that produced, it was very hard to produce this protein, it's a trick. So Camila went to John's lab and make this very good, this DECNU2. Camila also is here now, she works with John Adams. I think after the production of DECNU2, she decided to move to falciparum because it was so hard to work with Vivax. She's here working with falciparum. So what Camila showed that these people, we showed that these people that has high titer of inhibitory antibodies in the Amazon area was the people that has also high titer of antibodies against DECNU2. So also need to show this can inhibit the invasion of Plasmodium vivax. So the place matter, right, Delio? So Flora that works in my lab and Francis went to Porto Velho to work with Delio, and they make a short-term Plasmodium vivax culture that DECNU2 can inhibit the invasion of Plasmodium vivax. So because we have this DECNU2, I think it was Nunu that said sometimes you have inspiration, science needs inspiration and money. If you don't have money to go to the Amazon, let's go to the outbreak because it's close, you can check memory of these people. So we decided to return to the outbreak and check if that people that do not have more antibody, many years later, single Plasmodium vivax infection, if they still have memory B cells against DBP. So it was a short outbreak case that people had symptom and blood smear positive. In no case, no blood smear, no symptoms. Okay, whoops. The person problem. Here. No, here. Okay. So this is a BCL spot, it's very few. You can see this million secreted cells per million. You can see that, I hope that convince you these people have some kind of memory. This is different from these non-exposed people. These are the people that moved to the outbreak after the transmission. So they have these memory B cells. Okay. Differential gene expression, so that probably we have something different here. No, I think that I have to go here because I'm difference between case and non-case. This is more gene related to the immune response, and they are more non-specific response. Seems they have difference, but we have been a hard time to purify these cells because these people was a single malaria infection many years ago. So we have different tetramerase to try, right, Flora? Very hard. Now I start to make collaboration with Scott and Chet from UGA and Scott from Penn State University. So the idea is to make this decano multimeric to try to isolate these cells. So we are, for this we have been trying to use this system of these self-assembly protein units, subunits. So this Scott has been making this spike age express antigen for POLI and Chet for Sinomoj. And now we are doing this for Vivax. I think Flora just finished, right, Flora, to produce only, we will make this not only for DECNO2, for other P-Vivax antigen, like from MSP1 for Irene Soares. So the idea, this is much more bright, the different colors, green, red, and blue, so you can try to fish the cells. So we are in this time in this year. Moving to the endemic area, we have been doing this follow-up in a small community that's about 2 hours from Manaus, about 500 people. This is called Rio Pardo. You can see there is a stream here, the stream of Rio Pardo. So there is people that live around of this stream that in Brazil called Igarapé. And also people that live outside in this unpaved road, that's called Ramal. And these people that has this transcend immune responses, inhibitory antibody response, are more related with the Igarapé because they are have been more exposed for a long time for this malaria transmission. Just to illustrate that, different of the other studies in Brazil, this area, most of the people are native from the Amazonian area. So you can confirm this by the ancestry. You can see here in Brazil, we are like a mixture of European, Africa, and Amerindia. And you can see here that most of this have about 40% of Amerindia. When you check the Duffield genotypes, we know that in Amerindia they predominate more the allele A in blue here. So when you increase the proportion of Amerindia, we increase the allele A and decrease the allele B, that's in green, that's more related to European. And also we saw that, also Marcelo showed this before, that allele here, allele A here is associated with some kind of clinical protection, and allele B more susceptible to the infection, so we confirm the same results. I also would like to check if what's HLA class 2 makes some difference in this good and bad antibody response. So we make this a collaboration with Douglas Pires and David Asher from the University of Melbourne. So they help us to identify some difference in peptide bind growth. And you can identify, we found some allele groups that can explain good antibody response and poor antibody response. Not only good and bad, but also people that has more inhibitory antibody response, and then some allele groups that associate with the persistence of antibody response. So I'm not going to do this today, but people make a difference. When I was checking in the literature, you can see that, in fact, who is good response for this antibody response against malaria Also, it's good response for the virus vaccine like hepatitis, influenza, and the bad, also bad for the conventional. So people make difference. Okay, so the idea of our cohort that started in 2008 and was to oops, no. Is go up in different period of time. Went in periods of high transmission. Low, for example, he was 2 years without transmission, and sometimes we have the inspiration, but no money to go there, so arrived there, there was not so much malaria. In the last 4 years, I do like to say this, there is no, of course, the COVID, we have to stop some field work, but in the last 4 years, there is no more case. So all the patients, the individuals that we have here, they don't have circulate parasites anymore. So by different protocols of PCR. So this long cohort allowed us to classify these individuals at the end, like persistent responder or elite responder, temporary, that's not help so much. So I make some difference here in persistent and no responder. So, and this is the people that has inhibitory antibodies against different strain, like Decino-2, so good to show this. Just to illustrate, it's not so much people, you can see the little responder after so many years, it's like this, persistent PCR negative. And you can see that also Decino-2, there's a more persistence of antibody response that different allele of this is one that's common in that area, so go fast. So we are thinking at that moment, let's go to take the cells of these people, stimulate this PBMC with Decino-2 and DBP, and check what they produce, what cytokine they produce. I hope to convince you that it's very different, the persistent responder for the non-responder. And for the non-exposed. So you can see that we have increased a lot of pathway here, Th1, Th2, Th17. But it was very interesting, for example, if you take the pro-inflammatory, pivotal pro-inflammatory cytokine like gamma interferon, you can see gamma interferon is higher here, and IL-6 and 10F, that's more associated with immunopathology, is very decrease here, increase there. And also, I think that was very interesting that we found here a lot of IL-4. I think my former advisor, Fidel Zavalli, will be very happy that you find IL-4 here, because many years ago we showed that IL-4 was critical to make the crosstalk between Th1 and Th2. And now also there is a paper, I think, from the last year, behind everything that you know about the IL-4, the class switch, blah, blah, blah. There is some people that show cytokine is too much. I'm parasitologist. But what I think that was important, I forgot. 1 minute, okay. What was important that there is a paper from the last year that showed that IL-4 seems to be essential to keep the memory B cells in the germinal center. So, can be something interesting here. And also, I cannot also show this. Oops. There, in this control human malaria infection, a paper from the last year, they show what they showed that was associated, a strong predictor of protection was gamma interferon IL-4. Just to illustrate that this is different a little bit of the what asymptomatic that, for example, Greg, the very good Greg that's here, and Liz Antonelli, because in the asymptomatic they have a PCR positive. So they found they have the gamma interferon make a difference, but they don't found, for example, IL-4, IL-17, they don't have antibodies. So we have a lot of antibodies here. So the other question that to make this, perhaps these people only have antibodies, these huge antibodies against DBP and inhibitory antibodies. Let's go to check what they have antibodies against Plasmodium antigen. So we took this bioplex detection with Liz and Greg. That's a panel of about 16 Plasmodium vivax blood stage antigens. You saw these people have a lot of antibodies. So malaria is associated with protection. right? I will not go to this detail. How's the cell that's produced? Oh, there is a poster from, oops, it's not to look because, oh, okay. A poster from something that is more detail about this. Maria Fernanda make this, so Greg is helping us to understand, now start to understand what's a lot of subpopulation that's involved. This, I think, immunology in the last few years make like a boom of a lot of subsets and heterogeneous in B and in T. So what we found, I think Maria Fernanda presented this here, we have a lot of subpopulation that we identify, different follicular T cells. Everybody knows that CD4 T follicular T cells shape the B cells immune response. So we found the Th1, Th2 to H17, another subsets like this T cell that is supposed to be follicular, the CXCR5, but they don't have all the marks for follicular, and also B cells express this. There is a paper that suggests these cells also can be important to keep the reservoir of memory B cells in the germinal center. The famous Tibet girls that can, atypical CD4 that can be good and bad girls. This, I think this case good girls here. And also the atypical cells. There is a paper from a paper recently from the Shotong in Thailand that she showed that in a good malaria infection and convalescent, they found atypical Tibet cells that produce inhibitory antibodies that can inhibit DBP dark. But most of what they found was allele specific. Perhaps our are not more allele specific. So we have a lot of things that going on here. I don't know who, for this generation of this long-term antibody response, if this Vivax is sick game of the Vivax, if this can be stimulating bone marrow. Fabio tell us if you believe in Matthias, if there is parasite in the spleen, the bone marrow. But all this is long-lived plasma cell memory B cell. I think there is something in there. So I would like to thank OSB. This was. I will not go on this, just to take home. This is Brazil time for election. I will ask vote for DECNU 2, but not anymore, because John and I are convinced that's a very good tool to understand the people that has protection, but this is not to go to vaccine yet. It's a lot of tricks. So it was a wonderful tool to understand the immune response. So I will ask vote for another one. That's a secondary erythrocyte bind protein, that's EBP2. There is a poster here. And at least I convinced Santuza from CT Vacinas, or Ricardo, that this is they should be good. So Santuza believes us. I hope that's I'm not wrong. And she's trying to make a RNA vaccine with EBP2. And they are not cross-reactive antibodies. I think they are co-acquired. Hard time, right? Flora believe in this, that how to break. It was a very hard time to try to understand these B cells and purify. Now I hope with this Scott, this guy cage, you can make a cage to take this, these B cells. All they collaborate in all our students, in the field, and John that was very supportive. Thank you very much. A lot of reason to go to Belo, including this new stage.
Thank you very much, Luzia, for your very nice talk. Agora eu gostaria de chamar aqui, com grande satisfação, o professor...
So now I'd like to invite Professor Dr. Ricardo Gazinelli, full teacher at the University of Minas Gerais, researcher fellow in Fiocruz, Minas. He's trained in veterinarian medicine, also in immunology, and a postdoc in the United States. His trajectory is also marked by contributions to parasitary diseases and the development of vaccines against neglected diseases, including leishmaniasis and malaria. He's the coordinator of the ENCT vaccines and co-founder of CT Vaccines in UFMG Fiocruz. Please, Professor.
Boa tarde a todos.
Good afternoon, everybody. Not sure which language I should speak. I will try my English.
Good afternoon. First, I'd like to thank Carol and Danielle. I think it's a really great meeting. Really, congratulations. It's fantastic. The host from the beginning to the end, social our program, so congratulate. And thanks, Rodolfo, for the introduction. I'm gonna give a first 4 or 5 minutes. I just gonna talk about the center, the National Center for Vaccines, which is a recent institute at the Federal University of Minas Gerais, and how we got, and then we'll talk 50 vaccine. Today we are become this National Center, Vaccine Center, which started with the Technology Center for Vaccines from UFMG, and it's a partnership between the Federal University of Minas Gerais, the Technological Park of Belo Horizonte, the Fiocruz Minas, and we also recently become a center for RNA vaccines, which is from the Ministry of Health, both the Ministry of Science and Technology and Ministry of Health, our main funders. And here is our building under construction. So hopefully next year I'll show the building already in operation. So this center has the main mission is try to bridge the studies done at the university and bring to the industry, a little bit different from Biomanguinhos and Butantan, which is primarily focused on the production of vaccines for the Ministry of Health and for the National Program of Immunization. Our idea is to bring ideas, proof of concepts of the university to carry and develop prototypes and vaccines that can go to phase 1, phase 2 trials. And we're very proud because we got most of this funding during the pandemic to build the institute and to do a lot of our research in a government that was an anti-vax government. So we feel that we were very convincing. So the story, we received funding to develop a vaccine for COVID-19, and everybody was working with Spike. And we said, well, we should do something different because, of course, we cannot compete with Pfizer, Moderna, and so on. So we decided to work in a vaccine that was a T-cell-based vaccine that was not really, no one really focused on that. At the beginning, it was very polemic, but when we got, after all these variants start to emerge, maybe the idea was not that bad. So we used the part of the spike and then used the nucleocapsid protein from the COVID, which is very conserved protein in the various variants. So everybody asked, why vaccine for COVID? Of course, there is the continual emergence of SARS-CoV-2 variants. In Brazil, the updated vaccines, they always, the circulating variant is no longer, it's a new one. So they are always late. Of course, there is the immunoimprint, so the levels of antibody to the new variants are not very high. And the RNA vaccines require sophisticated cold chain for distribution, and they can reach many areas of Brazil. There is still a demand for COVID vaccine by the Ministry of Health. And of course, we also had fun to do this work. Some of the ideas that the current COVID vaccine, they induce low levels of neutralizing antibody which match to a specific variant. They quickly decline in about 3 months. And the idea today that maybe the T cells are really the most important arm to prevent not infection, but to prevent moderate and severe disease. And of course, the variants are escaping the neutralizing antibodies, so I think the T cells can be really useful. So I think I need to go faster. But so this is a result, oops, I like to show, which is, that's a preliminary result that we obtained in a phase 2 clinical trial of the vaccine. There are not enough patients, but we got really higher protection. P was not statistically significant, but we're using these results to convince Anvisa that we should move to phase 3. And we have here is the cumulative cases of COVID in individuals immunized with our vaccine versus the Pfizer vaccine. And here are all the variants that we got. Anyway, so one important thing of this program was really that we learned the roadmap to develop vaccines from the scratch, from the conception, going to approval at the Anvisa, which is our FDA. And then here is a picture of the first volunteer being vaccinated with our COVID vaccine. And we have concluded phase 1, 2. And so we now are applying this to other vaccines, and the malaria vaccine is the second one most advanced. But at the end, our vaccine is variant-proof, at least in the animal model. The logistic of distribution is much easier, is stable for 24 months at 4 degrees. It's cost-effective, and it's fully developed in Brazil. So here are some of the vaccines that we are working on. Many of them are for neglected diseases, and most are protein. But now we have a group of people that are also working on nucleic acid vaccines. And here is the stage of development, and malaria is the next one that I think we're starting to, planning to start the phase 1 clinical trial this year. You have seen the talk of Irene, so it will make much easier to make this talk. But we're claiming that we're starting to test what would be a universal vaccine for Plasmodium vivax malaria. So I can skip this, but there are 10 million people infected with Vivax every year, no licensed vaccine, and rising resistance to drug treatment. Also, this vaccine, I think you have seen this various times. They have a repetitive units of the 3 main variants of Vivax, VK210, VK247, and P-Vivax-like. You know all about that. It's a CSP protein that covers, so it's an anti-infection vaccine. So this vaccine was then produced in good manufacture quality. In general, this is a 25 KD polypeptide. In the gel, it looks like a 50, it's a dimer, and also gives you 2 bands, but there is a small, one fragment that is more few amino acid less, but in all preparations, they are kept identical, so which is important for the approval of the FDA here, the 2 bands. They're produced in Pichia, Pastori, and it's very good. I think we get 3 grams per liter of production. We have done all the primary sequencing, so it fits well with the plasmid sequence as expected. We have done all the characterization of secondary structure, tertiary structure. It's very stable, even with when you heat at very high temperature. Normally, the secondary and tertiary structure are preserved. Here, we have done some stability tests, and turns out the protein degrades very rapidly if you leave in solution. So we decided to use a lyophilized protein, that's this protein that contain, and use as an adjuvant the MF59, which is a squalene-based adjuvant. And to make our tests, we use the vaccine at 25 microgram per dose, 50 microgram per dose, and 100 microgram per dose. So here is the tests in mice. Here's the protocol. Priming, second boost, first boost, second boost. Well, again, priming, first boost, second boost, and measure the antibody. They're basically very similar levels of antibody using the different doses. And for the protein, and then higher levels of IgG1 compared to the IgG2C. And again, for all, induced nice levels of antibody for all 3 variants, both total IgG, IgG1, and IgG2. This I'll leave, I'll leave this to Luzia to explain for us, but if you look, the main conclusion, I can't make all those conclusions about BET cells either, but we conclude that the vaccine induces a response that's more biased, what they call Th2 response. We see IL-4, Luzia, here's IL-4, IL-5, IL-10. There is some interferon, but it's not that high, so which match with the idea that you have high levels of IgG1 in mice. This is an experiment for protection that we did with Rogério Amina at the Pasteur Institute, at the Pasteur Institute. Here are the 3 doses. And here's the level of protection. Non-infected mice were 100% protected at the dose of 50 microgram, 85% with 100, and about 75% with 25 micrograms. Here's the log of parastemia. You can see here no parastemia at all. Here and here 2 mice and 1 mice. And here is the percent of parastemia again. So we have a very good protection. And what I heard from Rogério is that not very common for all vaccines is tested to have 100% protection. These are rodent parasites that express the, I think is the 247, 210, right? 210, 210 antigen from the 210 strain, so, which is the most commonly distributed. We then did all the safety toxicity test, where you do at different days, and also the recovery group, and we look for the antibody response. We use mice, B6 mice. We have very nice antibody response. And this is not very intuitive graphs. It's a PCA. I don't expect you to understand the graph, but what it tells you is that there was not a lot of variation of the different parameters, hematological, biochemical, coagulation, and weight organs if you compare the different doses 2 hours after each dose, and also in the recovery time. So in conclusion, the vaccine was safe in mice, non-toxic and safe. We did the same experiments in primate rhesus. Again, the vaccine was immunogenic and induced IgG, IgG1, and IgG. We look just to IgG, to total antigen, and to each one of the variants, to VK210, P5X-like, and VK247. They're all immunogenic, or meaning if we immunize with the chimeric protein, we get a response to each one of the variants. So saying that the vaccine, and again, the same graph, type of graph showing that there is no any pronounced side effect that was outside of the limits that are acceptable for hematological coagulation and biochemical parameters. And this was using the higher dose, which is 100 microgram per dose. So finally, we did all these stability tests, evaluating the 3 doses. We went now for 24 months. We did all physical, chemical, microbiological, biological tests. This is in Portuguese, I'm sorry. Some of it is a mix, Portuguese-English slide. So we did the stability test at low temperature, at high temperature. Anyway, there was no major change for if you keep the lyophilized and the adjuvant at 4 degrees for 2 years. So we think it's a very stable and good vaccine to distribute in areas that you don't have much. Here was an experiment recently done by Maisa showing that the sera from mice immunized with the chimera or primates, they light up all the sporozoites, showing that they are recognizing the sporozoites. And now we're doing tests with her and Rogério to show the cytotoxic activity and also the invasion blocking activity in Porto Velho. So, in conclusion, it's a very stable, we obtain it in a very high degree of purity and it's very safe. We're now designing the clinical trial. I'm going to go fast. We're going to test 3 doses and 16 patients. And the main focus would be safety, but we also will look for some immunogenicity. And here is our timeline. So we expect to do the phase 1 in 2026. Phase 1b/2 would be, which will be done in Porto Velho, maybe Porto Velho and maybe another site in the Amazon, which should be about 2027 and 28. And if everything goes right, we go then to start the phase 3 in 2029. Here's the initial announcement to start recruiting patients, volunteers for the phase 1 clinical trial. And here is the group of CT Vacinas, which is today at the Technological Park. We have not moved to the new building yet. Here's our scientific committee, which are the PIs that run this institute. And here is the people that really contributed to this project on the malaria vaccine. I, of course, have to give a lot of credit to Irene that started the whole thing in her group. Rogério Amino, that has been a great collaborator in the last years to test some of the bioactivity of the vaccine. Maísa Indélio in Porto Velho with experiments and also helping to design the clinical protocol. Santuza and Elton at CT Vacina that coordinate lots of the control quality studies and the clinical trials. And Ana Clara and Ana Luisa that have really contributed a lot to lead these projects at CT Vacinas. Our main funders are FAPESP. For Irene, I'm not allowed to ask money to FAPESP. The INCT Vacina, FINEP, and again, the Ministry of Science and Technology and Ministry of Health. And thank you for your attention.
Thank you very much, Ricardo. Now we are open for questions em português, inglês e portunhol. So,
That was a really fantastic section, really. Sylvia, just 2 questions. Yeah, I know that. Oh, yeah. Yeah, I know, because this trick is for the other side. Yeah, yeah. Well, the first one is, when exactly you collect the B cells from the patients? Or how long for your assays? And the second one, you show some very interesting data that you have low binding but high killing of the parasite. What do you think? What's the hypothesis that you think is going on?
Okay, so the first question was when we collected the samples. So for this particular cohort, which was the one that was available to us, we collected samples when the people were sick and 20 days after treatment, 28 days after treatment. Okay, and then we pulled the sequence, the two sources of cells, and then tried the whole thing. But we are sort of we wrote another protocol with Maísa just to make we are going to try it again. Okay, in people that actually had like malaria, we were 100% sure that they had malaria, so we go to the CVAP files, and then we want to get people that had different events of malaria, but at the point of collection are free of the disease. Okay, so this is the second round of this thing. And then about so one thing that we know, Fabio, is that this repeats of the CSP, they are not very well organized, right? That's why, for example, we don't have a crystal structure with the antibody binding, because they sort of move around. So what I think is happening is pretty much like these antibodies that don't really bind that well, they're probably binding in other areas probably of the repeat that can sort of interfere with the interactions.
Okay, if nobody will ask, may I ask?
Yes, please.
Just Sylvia, congratulations, a lot of work. to produce the human monoclonal. But did you guys produce this against the acute malaria infection? So what's the idea? Are you planning to produce a monoclonal against people that have some kind of immunity against malaria here in Brazil?
It's really hard to hear.
Microphone, please.
Cannot hear. You cannot hear. She's just translating to me what she asked. Okay. The question produce the acute infection. Yeah, so that's exactly the second part. So for CS, I think, for example, every time the person is in the acute phase, for example, I don't think there's much CS circulating, right? Because it's already the parasites are they go from the skin to the liver very quickly. So I do believe that these people that are actually infected at the moment, their response is more to the infections that they had before. But this is something that Michelle was always telling us, like try someone that really had lots of infections, and maybe they have better B cells that are more, let's say, mutated and maybe higher affinity. So this is something that it took us a while to set up all the protocols, to get all the approvals, but we're moving, right? So yeah, so we're getting there.
First of all, congratulations to all you four. Really great lectures. And my question is for Silvia again. I was interested because there's a lot of new ideas to produce antibodies for passive immunization. And do you think you can actually produce antibodies with the IgG3 backbone, so they are more cytotoxic and maybe induce more cytotoxicity for the sporocytes? And you mentioned that you are going to do an experiment in vivo. Do you think the human antibodies may be a problem to use in mice, or do you think it's okay to use them in mice?
Okay, so the first question was about the IgG3. So this is a platform that you can basically add any heavy chain to your variable region, right? So you can very quickly and very rapidly swap the constant region. So you can do it. That's no problem. And the way we did this experiment, for example, we didn't. So in the beginning, like I generated a few antibodies against dengue and against Zika. And for those, we selected B cells that were memory B cells. So we stained them for IgG on the surface. For these particular experiments, we didn't, because we wanted to get like all kinds of B cells that were specific for the bait. So we could then go back and see if they were IgA, IgG, and IgM. And Mariangela did the IgGs, but did the IgMs also, right? IgM. So we can basically, like, if you sort only for the bait, you can go back and find what was the subclass of your antibody. And the second question was about the mouse. Oh yeah, in this case, you see, so obviously this is a human antibody. The mouse eventually is gonna respond to the human part of to the antibody, right? But this assays, we're going to be very quick, right? We're going to basically inject the antibodies, give them the challenge, and then wait like 6 days to see if the parasite escapes. So I don't think we're going to have enough time for the development of a response. But that's a worry if we're going to use this as a passive immunization in a mouse. But in humans, I hope it works. It should work.
I have a question for Professor Ricardo. I think you got better survival for. You got better survival rate for the intermediate concentration. I was thinking it could be due to very high inflammatory response to the higher antigen concentration. But I'm not sure, did you also get better parasitemia control for the intermediate antigen concentration? Or?
You're talking about the mouse
yes,
experiment.
Mouse data, yeah.
Yeah, this experiment was done once, right? So we have 50 micrograms, there were no parasitemia. 100, we have 85, I think. So it may be true, but also it's possible that if you do a second experiment, maybe a little bit different. But what's important is that we have done 2 experiments, and in both, in all 3 different doses, we got a very good level of protection. And Rogério can really tell you, because he has a lot of experience on that. So we can conclude that the vaccine induces good level of protection on that model. Yeah.
Thank you.
Hi, thank you. Very interesting. So the first question I have is for those involved in the Vivax vaccine that has the 3 components. And I was wondering, how was the order of those 3 different strain, the repeat components chosen, and whether or not that makes a difference? So if you put the Vivax like before the other ones, or, how was that decided? And then the second question is for Patrick. I thought PFS 230 was really variable. And so I was wondering, do you know if the vaccine is more effective against one part of the transmission cycle versus another? What's happening with PFS 230?
Yeah, for the first question, I think that, we also saw this being, and from our results, the epitope, which is the last one, normally gives a better response, which is the most dominant variant, that's the 210K. But we don't know if we change this because it's a better epitope or because it's on the better position. And also there is a really small part that's conserved in all 3 variants. That's the N, is the peptide that comes on the end part of the, and that's the same for all of them, and supposed to be a very important epitope. So it would be shared by the different variants. So I agree. It should be. We should test that, but we don't have
As far as the PFS 230D1 goes, it does have polymorphisms in it. The immunogen itself, actually, a lot of the face of the protein is a target of antibodies, so there's multiple epitopes on it. That's fortunate. Half of it is actually occluded in the native antigen by this domain 2, downstream domain. So none of those antibodies are functional, but on the exposed face, there's at least 5 functional epitopes that we've defined. Neeraj Tolia, who's my colleague at LMIV, so he used the antibodies, the monoclonal antibodies we made to define these epitopes, and then showed that only one of them was influenced by a polymorphism in it. So it is something that we need to track over time. There are polymorphisms in PFS 230D1, but with all the existing sequences, we know we could only find one polymorphism really that had an impact, and that was on only 1 out of 5 functional monoclonal antibodies that we had.
So thank you all for the presentations and the discussion. We will move to the next section.